My particular research has been on using pathogens, insect pathogens, as models for understanding, not just in insects, but in us as well, how pathogens evolve, how they get a narrow host range. What does it take for a pathogen to evolve a broad host range? And particularly because there's issues with human disease, why do some pathogens able to jump from one host to another? Malaria is, of all the insect-borne diseases, insect-vector diseases, that's the one that kills the most people. It's currently killing about 1 million people a year, mostly children, mostly in sub-Saharan Africa. There's been a huge effort to try to curb malaria. Every time we've created a new insecticide, a new method of mosquito control, which seems innovative, the mosquitoes have always been able to out-evolve it, outflank it. So we need another new method, just another new method, and biological control seems one way we can go about it. Not use chemical insecticides, but use biological insecticides. You've got viruses, you've got bacteria, you've got fungi. The best method for mosquitoes in particular is a fungus. The fungus acts like a contact insecticide. It will attach to the cuticle. It bores through the cuticle, which is the skin of the insect, gets into the blood of the insect. Our first approach was to put a toxin, a gene encoding a toxin, into the fungus. The gene came from a spider. We can get the insect dead really quickly. You create a lot of selective pressure in those insects, they'll out-evolve the fungus. So, new strategy, let the mosquito live, selectively kill the malaria. We took a gene, a human gene, encoding a human antibody. We took a couple of other genes too, which would kill the malaria, and we put those genes into the fungus. We use those against the mosquito, and it produces the anti-malarial in the blood of the mosquito. Within a couple of days, the mosquito is basically cured of malaria. With our new molecular techniques, we could produce a pathogen, or a range of pathogens, which could recycle through pest populations, more or less permanently suppressing them. And we could engineer pathogens adapted to particular individual environments as well. We can fine-tune these things now to a considerable degree. We've got the genomes of these different things. We know all their moving parts. We've got a whole list of different promoters, which are the on-off switches for these genes, to selectively get these genes to be expressed at different times and places exactly when we want them to be expressed.